Facial skeleton. The facial skeleton comprises the facial bones that may attach to build a portion of the skull. The remainder of the skull is the braincase. In human anatomy and development, the facial skeleton is sometimes called the membranous viscerocranium, which comprises the mandible and dermatocranial elements that are not part of the braincase. Structure. In the human skull, the facial skeleton consists of 14 bones in the face. Variations. Elements of the cartilaginous viscerocranium, i.e., splanchnicranial elements, such as the hyoid bone, are sometimes considered part of the facial skeleton. The ethmoid bone, or a part of it, and also the sphenoid bone are sometimes included, but otherwise considered part of the neurocranium. Because the maxillary bones are fused, they are often collectively listed as only one bone. The mandible is generally considered separately from the cranium. Development. The facial skeleton is composed of dermal bone and derived from the neural crest cells, also responsible for the development of the neurocranium, teeth and adrenal medulla, or from the sclerotomy, which derives from the somite block of the mesoderm. As with the neurocranium, in chondrichthyes and other cartilaginous vertebrates, they are not replaced by endochondral ossification. Variation in craniofacial form between humans is largely due to differing patterns of biological inheritance. Cross-analysis of osteological variables and genome-wide SNPs has identified specific genes that control this craniofacial development. Of these genes, DCHS2, RUNX2, GLI3, PAX1 and PAX3 were found to determine nasal morphology, whereas either impacts chin protrusion. Additional images.